Welcome guys, in this video tutorial we are going to talk about NET exam, which are the most important topics for the ICAR and for the ARS NET exam. What you have to read from each and every module, and what are the most important topics that is majorly asked in this exam. So in this video we are going to talk about each and every module and their topics that is most relevant to the exam. So the first module, that is cell structure and function. So from this module you have to learn about different cell organelles and their structure and functions. So the structure and functions of cell organelle. Cell organs, their structure and their function. Structure of each and every cell organelle and their function. Second most important topic for this module, that is the cell division. You have to learn about cell division. What is the most important protein for the cell division? That is cyclin and CDKs. So different cyclins and CDKs that is involves in the cell cycle and cell division. And for the cell cycle, how it regulates to the cell division. What are the most important cyclin CDKs for the cell division? Cell cycle is most important topic for this module. Now another most important for that, regulation of cell cycle and protein secretion and targeting. So the targeting organelles, you have to how protein is going to be targeted from the one organelle to another. So that is most important. Protein targeting is the most important topic for this module. Now if we talk about the second module, that is biomolecules and metabolism. So for that you must have to go through the major biomolecules and their roles and their structures. So the major biomolecules, that is carbohydrates, proteins, lipids and vitamins. So their structure and functions is most important. Now another most important, that is the about the plant and animal hormones. Plant hormones is the most important and their precursors, hormones and animal hormones. The precursor for each hormone that is most important in plants, for the auxin, gibberellin, cytokinin, ethylene, abscisic acid. So amino acids are the precursor for enzymes. So you must have to learn about the precursor for everything, for vitamins also, for hormones also, you must have to understand. Second most important for this module is you have to understand the metabolic pathways. Metabolic pathways. For that metabolic pathways, you must have to understand the enzymes involved in the metabolic pathway, as well as interaction and ATP calculations. So there is most important for this module, that is calculation of ATPs for glycolysis, for citric acid cycle, TCA cycle, and regulation of glycolysis is most important. Regulation of TCA cycle is most important. Electron transport chain. What is the site for this metabolic pathways? That is most important. Where it occurs for this organelles that is involved in this metabolic pathways. Suppose electron transport chain. It occurs in the mitochondria organelle. So that is the site for each and every metabolic pathways, and their interaction between these two pathways like glycolysis, TCA cycle, then electron transport chain. This is the interaction, how their pathways is going to interact to each other. Everything that is supposed from the glycolysis to TCA cycle, there is interaction of pyruvate, conversion of pyruvate to acetyl CoA. How it, what are the enzymes and what are the molecules like TPP that is involved. That is most important for this module. Now for the third module, that is about enzymology. For the enzymology, you must have to understand about the classification of enzymes and their examples. Suppose catalyzed enzyme is belongs to, they are asked about this type of questions. So enzymes, for that module, classification of enzymes, that is most important. Different purification techniques for enzyme, that is you can read from the Leininger book, as well as you can read from Wilson and Walker book also, for the purification of enzymes. Second most important, that is the enzyme activities. You must have to go through the enzyme activity. Then enzyme inhibition. What is the term you have to must understood about the Km, Kcat, then Vmax. What is the regulation of for these enzymes? What are the specificities, different types of specificities for the enzyme? You must have to understand this type of things. Just you have to learn about what is the Km and Vmax value for the enzyme inhibition, competitive for non-competitive inhibition. So you must have to learn about this type of things. You must have to go through the Michaelis-Menten equation for this module. From the fourth module, now we are talking about what is the most important topics you have to learn about from the fourth module. So that is from the fourth module, you must have to learn about the genetic organization of prokaryotes as well as for the eukaryotic organisms. Then you have to understand the different topics, that is the operons, the structure of operons, how they are going to be regulated. The most important, that is the leg operon, then tryptophan operon. They are asked some of the memory based question from this operon. Next, for that you must have to learn about the mutation. Mutation, you must go through the ones for the mutation. Then transformation, transduction and conjugation. Transformation, transduction and conjugation. It is most important for this module. These things only you have to go through once. What are the objectives they are asked from this exam? Suppose like mutation, spontaneous mutation, there is also induced mutation. You must have to go through the site directed mutagenesis. This and recombination is most important. What are the genes involved in the recombination process? How the recombination process occurs? Then different structure of chromosome structure is most important. Now another thing, that is what are the genes, that is family, gene family involves. And another thing that is most important is the exon intron. About you must have to go through once for the exon and intron and splicing process. For this intron and the splicing process, you must have to understood about the splicing process. From the next, that is the gene expression. For the gene expression, the most important topic, that is the central dogma. Central dogma of life. For this, you must have to understand about the replication, transcription and translation, and the post transcription modification and the post translation. For this, you must have to understand the enzymes for that particular process, like helicase, the role of helicase, the role of DNA polymerase. They are asked about the exonuclease activity, what is the 5' to 3' exonucleic activity, the topoisomerase 1, 2, 3. These things you must have to understand about this. Then transcription and the for the transcription you must have to understood about the factors, transcription factors, the sequence of transcription factors that is given in the Leninger book. You must have to go through once. Then formation of protein on the ribosome, there is the translation process, and the genetic code is most important also for that, and the transcription, translation, and the modification, and the operon concept is the also operon most important for this gene expression studies. So for the sixth module, that is the techniques. For the techniques, you must have to understand about the role of techniques. So why technique is used, for that different types of techniques, and most important chapter, that is the 19th chapter from D.D. Singh book. You can read this book. That is the easiest technique that is given role for each and every techniques, like chromatography used for the uh, separation of colors, separation of components, gel electrophoresis, like agarose gel electrophoresis, SDS polyacrylamide gel electrophoresis, polymerized chain reaction, the component used in the PCR, that is given by Carey Mullis, and it is carried out in thermal cycler machine. Then you have to understand about the nucleic acid hybridization. What is the role of why southern hybridization used? Western blotting is used for the detection of DNA and protein. And the next important that is about the monoclonal antibody and polyclonal antibodies, and the detection technique, that is enzyme linked immunosorbent assay, that is the ELISA, is also most important. For this DNA sequencing, what is the type of DNA sequencing, what is the method of DNA sequencing is most important, like PIO sequencing, first generation, second generation sequencing, that is most important. So this is all about the first 6 module, what you have to learn about this for to crack the ARS or ICAR-NET exam. I hope this video is helpful to guys. If you want to learn about different topics, you can comment in my comment box. I hope this video is helpful to you guys, and the next part for this next 6 module, and you can donate us to cruise biology. Thank you for watching. Thanks for watching. I hope this is helpful to you guys. Thanks.